Hi, I'm Heather, and this edition of the Longevizine is for the week of June 1st, 2026. A research team at Mass General Brigham fed more than 11,000 transcriptomes into a single model and asked a question that, now that it's put into words, is obvious. Does aging look the same wherever you find it? Their answer, published this week in Nature, is that to a striking degree, it does. A transcriptome is the full readout of which genes a cell is actively expressing, the working state of the machinery, rather than the blueprint, and across tissues, and across mammals as different from one another as a mouse and a person, the same families of genes shift in the same directions as the years accumulate. Aging, on this reading, is not 1,000 unrelated breakdowns scattered through the body. It is a coordinated pattern, a signature, and signatures can be read. The paper, Universal Transcriptomic Hallmarks of Mammalian Aging and Mortality, Tyshkovskiy, Gladyshev, and colleagues, Nature, DOI 10.103/S41586-026-10542-3, did two things. First, it identified clusters of genes that move together as tissue ages, co-regulated modules that recur whether you are looking at liver or brain or blood, and that hold their shape from one species to the next. The persistence of those modules across such evolutionary distance is the real surprise. It suggests that whatever aging is, a meaningful part of it is conserved, written into a layer of biology old enough that a rodent and a human still share the handwriting. Second, the team turned that pattern into a measuring instrument. They built multi-tissue clocks that estimate biological age and predict mortality in both short-lived and long-lived animals, and they released the underlying tool publicly as the Transcriptomic Age Calculator Online, or TACO, so that any researcher sitting on RNA data can score how old a sample looks, regardless of which tissue or species it came from. The temptation is to read Universal Aging Clock as, "We have found the dial." We have not. The most useful sentence in the entire announcement came from the study's senior author, Vadim Gladyshev, who pointed out that in humans we still do not have a single intervention proven to extend lifespan. What the study delivers is not a lever, but a far better ruler, one that can tell you whether a drug, a diet, or a genetic change moved an organism's biological age, and in which direction, with a precision that earlier clocks could not manage across many tissues and species at once. The older generation of clocks tended to be trained on one tissue, often blood, and to read methylation rather than expression. A clock that reads the active transcriptional state and generalizes across organs is a different kind of object. It is closer to a thermometer that works no matter where you put it. For anyone running their own experiments on themselves, that distinction is the whole game. A measurement you can trust is the precondition for an experiment worth running, and most of the noise in the self-experimentation world comes from people changing 3 things at once and then reading their results off a bathroom scale. CGM, HRV, and a vibe check. A clock that scores biological age the same way across tissues is, in principle, the instrument that would let an N of 1 become an actual experiment, a baseline, a single change, a reread. The promise is that biological age stops being a vague atmosphere you sense in the mirror and becomes a variable you can perturb and watch. The work is early, the tool lives in laboratories rather than living rooms, and the gap between scoring age and slowing it is real and as yet unbridged. New insights in long COVID. The second development is smaller, quieter, and in some ways more instructive because it shows what happens when a tidy, mechanistic story meets a careful measurement and loses. For years, the leading explanation for long COVID's most stubborn symptoms, the fatigue and brain fog and low mood that linger long after the infection clears, has pointed at the brain itself. Persistent inflammation smoldering on, keeping the system in a state of alarm. It is a reasonable hypothesis. Severe acute COVID does leave inflammatory marks on the brain, inflammation is measurable, and it is a satisfying villain because it suggests a target you can aim a drug at. A team at the University of Turku decided to look directly rather than infer, and what they found complicates the picture. Their study, published in the Journal of Neurology, Tuomala, Iris, and colleagues, J. Neural, 2026, 273, 298, DOI 10.1007/S00415-026-13842-W. used TSPO-PET imaging, which lights up the activated glial cells that mark neuroinflammation, alongside MRI to assess structure and blood biomarkers of neuronal and glial damage. They compared 14 people with persistent long COVID symptoms against 11 healthy controls and 13 people with multiple sclerosis, a disease known to inflame the brain and included here precisely as a positive reference, a picture of what real, measurable neuroinflammation looks like on this scan. The long COVID group showed no sign of widespread brain inflammation relative to healthy controls, and markedly less than the MS group. The broad inflammation hypothesis did not survive contact with the scanner. What did show up is the more interesting part. In the patients with the most severe fatigue, anxiety, and depression, the glial activation that did appear was localized rather than diffuse, concentrated in the limbic system, thalamus, amygdala, hippocampus, the circuitry that governs mood, threat, and memory. The worse the symptoms, the more activity in the regions that handle emotion and stress, and not the diffuse, system-wide signature you would expect if inflammation were burning every room in the house. The researchers' suggestion follows directly from the geography of the finding. Patients carrying this burden may be served better by approaches aimed at stress and emotional regulation than by therapies chasing inflammation across the whole brain. 14 patients is a small sample, small enough that a few unusual individuals could tilt the result. This is a single imaging modality, and the wider literature on long COVID neuroinflammation remains split, with capable groups pointing the other way. What this study gives us, therefore, is not a verdict, but a reframing, a strong signal that the question may have been aimed at the wrong layer of the system. And that reframing happens to land where this work has always pointed, the nervous system as a regulator of state, the limbic and autonomic machinery that decides moment to moment whether the body sits in alarm or in repair. Vagal tone, heart rate variability, the felt sense of safety, these are not soft adjuncts to the real biology. In a body where the most severe chronic symptoms track the activity of stress and emotion circuits rather than the residue of damage, the regulation of state turns out to be the biology, not the wrapper around it. The messy intersection of quantum biology and warm, wet bodies. Quantum biology spent 2 decades being treated as an embarrassment, the warm, wet, and noisy objection, deployed like a closing argument. Delicate quantum states cannot possibly survive in something as messy and thermally chaotic as living tissue. That objection has been eroding for years, dismantled less by argument than by data, and 2026 is a year in which the field looks less like a fringe and more like a research program with conferences, journals, and a maturing sense of its own boundaries. The Frontiers research topic, Quantum Phenomena in Biological Systems, editorial DOI 10.3389/FPHOT.2026.1826400, gathers the foundational cases in one place. The near-perfect efficiency with which photosynthesis moves captured energy toward its destination, photon capture in vision, and the radical pair mechanism proposed to explain how migratory birds read the Earth's magnetic field, in which a single photon creates a pair of electrons whose correlated spins are sensitive to magnetic orientation. The task is to map where quantum dynamics actually do functional work in living systems, not to declare that they are working everywhere at once, which is a complete excuse to avoid doing the work or thinking. Thinking is hard. For specific systems, the evidence is real and strengthening. Photosynthetic energy transfer and avian magnetoreception are no longer punchlines. They are problems with serious data attached and serious physicists arguing over the details. For the more thrilling claims, the ones about microtubules and consciousness and a quantum substrate to the mind, the verdict is unsettled, and the reasons the evidence stays thin are themselves something to consider here. It is extraordinarily hard to measure coherent quantum states inside living tissue at body temperature. The instruments capable of even attempting it are young, and the clean, decisive experiment that would settle the consciousness question one way or the other does not yet exist. Thin evidence is not the same as no signal, and it is emphatically not the same as proof. It is an invitation to build better instruments, which is precisely what the coming month is partly about. 2 meetings in June mark the consolidation. The Quantum Effects in Biological Systems Conference runs June 3rd to 5th in Budapest, and the Quantum Biology, Cognition, and Medicine Workshop runs June 10th to 11th in Växjö, Sweden, embedded in the QIP26 conference. The titles alone tell you where the frontier thinks it is heading, from foundational physics toward cognition and medicine. Watching what actually comes out of those rooms is a better use of attention than any number of confident pronouncements about what quantum biology has proven, because the people in those rooms are mostly arguing about what it has not yet proven. And that argument, conducted in public and in detail, is how a field grows up. Seeing the pattern. Set the aging clock, the long COVID scan, and the quantum biology calendar side by side, and a single motion runs through all of them. Each one moves the explanation away from biology as parts and toward biology as information, biology as field, biology as pattern. The aging clock does not find a broken component to swap out. It finds a signature written across the whole, a coordinated drift in how thousands of genes are expressed, and it reads that drift as a single number. The long COVID study does not find a smoldering lesion to extinguish. It finds a pattern of activity in the circuits that set the body's emotional and physiological state, and it suggests the symptoms live in that pattern, not in any wreckage. Quantum biology, at its most grounded, asks whether some of life's most basic operations run on coherence and spin, on the relationships between states rather than on the discrete states themselves. 3 different research communities, none of them talking much to the others, leaning in the same direction. Let's be precise about what this convergence is and is not. It is not a claim that the 3 findings support one another evidentially. They come from unrelated fields and rest on unrelated data, and none of them needs the others to stand. It is a claim about the direction the questions are pointing. When a clock reads state instead of cataloging damage, when a chronic illness is found in a circuit's activity rather than in a lesion, when a serious branch of physics asks whether coherence does real work inside cells, the assumption underneath all 3 is that the relevant unit of explanation is the relationship and the pattern, not the isolated part. That assumption changes how you would even design an intervention. You stop hunting for the one broken gear and start asking what would shift the whole reading, which is closer to how the body has always behaved for the people who pay it close attention, and closer to what these 7 pillars have circled from the start. This is the same idea wearing 3 different outfits. A machine is a sum of parts, and you fix it by finding the broken one and replacing it. A score is a pattern read in time, and you change it by changing the reading. More and more, across domains that have no particular reason to agree, the body looks less like the first thing and more like the second. That is not mysticism dressed up as science, and it is not science apologizing for sounding mystical. It is what the measurements keep saying when you let them speak without forcing them back into the machine metaphor. The ruler that reads aging across species, the scan that relocates chronic illness to the circuitry of state, the field that asks whether coherence is load-bearing in living things. All 3 describe a body that behaves like something being read. And once you are willing to take that seriously, the question of who or what is doing the reading stops being a category error and starts looking like a research question with a long future ahead of it. The science and the mysticism were always the same thing, just using different words to describe the source code running the whole thing. What's next? The aging clock lives in laboratories. The long COVID finding is a reframing that needs replication at real scale before anyone changes a protocol. And quantum biology is walking into a June where the most valuable sentences will be questions. What you can do in the meantime is calibrate your attention. Watch the instruments rather than the headlines about the instruments. Notice when a scan disagrees with a story and give the scan the benefit of the doubt. And keep a little room for the possibility that the most rigorous current in this science and some of the oldest intuitions about what a living body actually is have started, slowly and from opposite ends, to point toward the same place.